Hello, interested everyone. Welcome to class 11 biology. In this video on chapter 13 photosynthesis in higher plants, we are going to discuss about dark reaction of photosynthesis. And specifically we are going to discuss about Calvin cycle. Right. In our previous video we have discussed about light reaction. And in light reaction we have seen the production of energy rich molecules like ATP and NADPH by use of light energy. Imba dinge de ngazu ki video na ngazu khartare light reaction ko lata imba the photochemical phase. This is khari produce chere plant ki chloroplast ki khari produce cheso by using light energy ATP da NADPH produce chigiwa. So ATP the NADPH the kharchini produce chigore the khala pheju chere la wi na dark reaction che to pheju chere. And the dark reaction ka to khari chungore. So let's see what is dark reaction. So dark reaction la zambala it is also called as the biosynthetic phase of photosynthesis. Light reaction, right? Light reaction nang khalo yo photochemical phase lagi yo. Dark reaction nang khalo ore biosynthetic phase la or. So in this particular phase of photosynthesis, what happens? Right, during this phase, there is synthesis of sugars, right, with the use of ATP and NADPH. So light reaction kabla ATP the NADPH so kyo deta dela pheju chigor. Dark reaction ka to kharche hai to pheju chigor la wi ba na sugar synthesize chayala, carbohydrate synthesize chayala. So ATP and NADPH, which is produced during light reaction of the photosynthesis, is used during dark reaction to produce sugars. Ta ming dark reaction it is a misnomer. The ming tata dego marislar. It does not mean that the reaction occurs in the dark. Synthesis of sugar, the reaction which produces sugar, right? It does not occur in the dark. The dark reaction, it continues when the light is not provided to the plant for just a little bit. And dark reaction the plant after that it will stop. The proof light may light reaction chungumare. Light reaction machung imbaina ATP the NADPH swetu mare. ATP the NADPH swematu imbaina and the dark reaction chungumar. The zambala dark reaction like minchtoa. The nagung kala yamena in the night time plan gi onda and carbohydrate swore lago marda. So it is said dark, the name is given as dark reaction, but what it actually means is it is not directly dependent upon light. Right, directly dependent upon light reaction light reaction photochemical phase in a dark reaction kab do any reaction the chungwala sugar produced chewala any light reaction khaji product the go ATP the NADPH go so indirectly light reaction the dark reaction the light latent there. So therefore it is a misnomer. Okay, teacher ta ngazu ki light reaction the dark reaction ki chungsa ki saja de law in by na ta ngazu chloroplast ki sopta de ha ko you know the structure of chloroplast. In the chloroplast we have got these stacks of thylakoid, right? Thylakoid mangbu chik shegyo de ngazu khale granum lagya, so the granum the chembu sunta wen de do. So granum nanglo yo ki thylakoid membrane nanglo la ani light reaction chungor, right? In the thylakoid membranes, right, light reaction occurs because in the thylakoid membranes the chlorophyll molecules or the pigment molecules are present, the photosystems are present, and it results in the production of ATP and NADPH. In the presence of light, the thylakoid membrane nalo yogi chlorophyll denzo kharchi gore ATP da NADPH so gore through light reaction. And ATP the NADPH the kharchi hechu the pheju chigre la wimbena for the production of sugar. And for the production of sugar, ATP and NADPH, the reaction the dark reaction. So this particular diagram, in this particular diagram, you can see the location of light reaction and the location of dark reaction. Light reaction occurs in the thylakoid membranes, or the membrane system. Dark reaction, it occurs in the stroma. Production of sugar happens in the stroma, because the enzymes necessary for the production of sugar is present inside the stroma of the chloroplast. Okay, so diche ta tiwa thangburda. What is the first product formed when carbon dioxide is fixed. Tanga jo kharla gore dark reaction kadu carbon dioxide pheju chini ani carbohydrates go sugars go reslarwa. Tinde himba ina. Carbon dioxide chilo ni ya theni. Atmosphere ne carbon dioxide ya nanglo la leni ani plant gi supu nanglo la yamena chloroplast nanglo la. What is the first product formed? Right, carbon dioxide atmosphere ne ya dus thewa da plant gi product thangsho kharsyo go reslar. What is the first product formed? So di tadu zambala di gala seri pa mangbu shuji gi experiment cheni ro kale shuji kan kunju gi kharis product thangbu kharsyo du ta ha khwa try cher. Di nanglo ne scientist nenga chishu ro contribution nenga chishu su ga jore la wen bana Melvin Calvin lad ro his name is Melvin Calvin, right? So Melvin Calvin gi kharchere he used radioactive form of carbon, right? C14 ladu zambala, it's the radioactive isotope of carbon. Koigi radioactive isotope of carbon pheju cheni koigi khachere algal photosynthesis trace cher koigi. He started his studies on algal photosynthesis. Algae la wimbana green photosynthetic organism ro. So algal photosynthesis study chedu zambala koigi khari pheju chere C14 radioactive C14 pheju cher. And then he tried to trace ani reaction khandi khandi nanglo la ani product khari form chi do carbon di khaw khol du do ko ta tu wor. And he discovered that the first carbon dioxide fixation product was a 3-carbon organic acid, which is called as 3-phosphoglyceric acid or PGA in short. Melvin Calvin gi kharla re carbon dioxide plant gi supu nalo dus ya lewa ta product thangsho khari chag dulare it is a 3-carbon organic acid. 3-carbon organic acid dikhare 3-phosphoglyceric acid la this dular. Here is the 3-phosphoglyceric acid formula, the structure they did. And he also figured out the complete biosynthetic pathway of sugar production. Sugar produced share jomarwe dikhogi complete cycle dikho ha gor cyclic form jora dicheni and the reaction jomarwe carbon dioxide ma fix cheni glucose soya gi reaction jomarwe tila kharla gore Calvin cycle ke la, because this biosynthetic pathway it occurs in a cyclical manner. Okay, so he helped in figuring out the complete biosynthetic pathway, and therefore koigi ming di kharla tobre Calvin cycle and cycle de ming la tobre. So diche, and later on Calvin gi kharsa Melvin Calvin gi kharla re carbon dioxide supunal ya dus lewa da plant gi supunal la carbon dioxide ya lewa da product thangbo di 3-carbon compound redula ro 3-phosphoglyceric acid redula ro. Now other scientists khonju ge tadu zambala khonju ge experiment chidu zambala they found that not all plants shemba changa ani 3-carbon compound sugum indus. Later on scientists found that not all plants had 3-phosphoglyceric acid as the first product after carbon dioxide fixation. Some plants they produce 4-carbon organic acid. Carbon shisheveki organic acid oxaloacetic acid or OAA in short as the first carbon dioxide fixation product. Plant khashiji harchigore carbon shisheveki organic acid sugore plant khashi harchigore carbon sumcheveki organic acid sugore carbon dioxide subuna lemada. Di induzambana plant nalo rigmanda ni chaswa C3 plant da C4 plant. So there are 2 types of plants, C3 plants and C4 plants. Why are they called as C3 plants and C4 plants? Because in C3 plants, right, the first product after carbon dioxide fixation is a 3-carbon compound called as a PGA or 3-phosphoglyceric acid. Whereas in C4 plants, right, the first carbon dioxide fixation product is oxaloacetic acid, which is a 4-carbon compound. So therefore, it is called as C4 plants. So khonsu nalo la pathway manda ni chaswa C3 pathway ta C4 pathway lani. So the biosynthetic process or the dark reaction is of 2 types. Dark reaction la rig manda near C3 pathway ta C4 pathway lani. C3 pathway la the sugi discover chere Melvin Calvin ki cherwa. So the imposonza Calvin cycle la isto. We will focus only upon Calvin cycle in C3 pathway. C4 pathway nalo la 30 manda yer. But main so the kharcha rule and C3 pathway the chart rule. So the into zambala before we go into Calvin cycle, another question is there. What is the primary acceptor of carbon dioxide? Carbon dioxide thangbo accept chingegi molecule di kharire plant gi suku nanglo la carbon dioxide ya nanglo zuwa na molecule khande gi carbon dioxide di accept chigore chiwo gu tang ro tang azu gi thanda khare ha go re atmosphere ne carbon dioxide ya zugor ro ani product thangbu khari form chigore la wi na 3 carbon compound called as 3 phosphoglyceric acid ta de carbon sum chigor do carbon dioxide nanglo carbon chigma do yo marwa so de la carbon dioxide accept chenge gi molecule carbon khazu chegyo jore la wi na thiwa ta wi na azu khari tensi gre carbon ni chegyo compound ji ingire samdi gro hajin na dela carbon chignya ni yo pare carbon ni chegyo compound ji accept chegyo wi na ani carbon sum chegyo gi pga di chagro but later on scientists they found out that the primary acceptor of carbon dioxide was a 5 carbon ketose sugar carbon nga chegyo gi compound ji ba hawkor it is quite surprising for the scientists. For the scientists, it took a lot of time for the scientists to figure it out. Right? Kajin lana mi mangche khar se dere carbon ni che jo compound che se derta uzu gen. Ine last de lokon so khar ha kore law in na carbon ni che jo marishas carbon nga che jo gi ketose sugar che reshas ti khar reshala na ribulose bisphosphate RuBP in short. Ribulose bisphosphate la di ha che gore carbon dioxide dia le ani ko khar so gore 3 pga di so gore 3 pga lana khare 3 phosphoglyceric acid. Okay, so dire tadi ming de zonenga chimburta ribulose bisphosphate lash to it is a very important name you have to remember, right? Ribulose bisphosphate is the 5 carbon ketose sugar which acts as the primary acceptor of carbon dioxide within the plant. Plant nang lo la carbon dioxide thangshu sugi nyagre accept su chigre la wi bana atmospheric carbon dioxide di 5 carbon ketose sugar chigi chigre sti kharre la wi bana ribulose bisphosphate or RuBP. RuBP ki full form di ribulose bisphosphate. You remember that, okay? Diche. Tang azu khare Calvin cycle ka discuss chayenda. So dinalo la 3 carbon compound form chigido. So let's first discuss about the Calvin cycle or the C3 pathway. Ra. Ta nyene de tanda nyene se usuerda. Calvin cycle it takes place in all photosynthetic plant, whether it is C3 plant or C4 plant. C3 plant imbainere C4 plant imbainere ro Calvin cycle la di plant photosynthetic organism sangma nal chungor even in algae ro ngazu gi khare green photosynthetic organism algae plant mai ba algae la choro din anglo le kharchi gore Calvin cycle la di chungor now Calvin cycle la gi cyclic reaction pathway choro din anglo la 3 stages na ngazu kopsha yogor for our own convenience, right? Aranzo sosogi jang lobjon chedu zambala ani aranzo convenient yuwa chedu lelo chen lobjon chedu he chedu jang thuye chedu ani Calvin cycle la jumare stage sum lo kopsha gya shayar har khare carboxylation, reduction and regeneration ngazo gi mitosis ta nazo gana jaimba prophase metaphase anaphase telophase lan jaimba dinangshi ngone cheno ko khare continuous process chigro ine ngazo dezen na yewa che go yumze khare law imba na susu conveniently ya bujene jang thuye chedu ha ko ye chedu ro din zambala Kelvin cycle de thumbusum la yewa che shayre ro stage sum la yewa che shayre stage sum dikhar kar re law imba na thangbo carboxylation che niwa dikhare reduction sumba di regeneration. Now during carboxylation what happens during the first stage of the Calvin cycle carbon dioxide jumara ta dikran loktem nangri mudirda atmosphere ne carbon dioxide dikhachi ore it enters the Calvin cycle carbon dioxide dia nanglo lewada khachire ribulose bisphosphate gi accept chiro so RuBP ribulose 1,5 bisphosphate lash to so this is a 5 carbon ketose sugar which acts as a primary acceptor of carbon dioxide accept cheni the exception kuni arrangement de dilangazu kharla or it is called as carboxylation. So carboxylation ka kharchi gore carbon dioxide combines with RuBP nyamdola combine chigor dilangazu khalare carboxylation. And what is the end result of carboxylation? Carboxylation results in the formation of 3 phosphoglyceric acid. Imba manzugi carbon dioxide nanglo ya fix chewa talam sa product thambu khar form chigore laimbe 3 phosphoglyceric acid. Therefore, it is called as C3 pathway as well. C3 pathway ke lagoro. Kajin carbon thangbu di kharcha ho carbon sum che yogi compound ji chagoro carbon dioxide ka ji product thangbu di. Ade ya usia. Carbon dioxide chik dine leso. Carbon chik de leso. ribulose bisphosphate la dina carbon nga yorwa so carbon nga plus 1 carbon dioxide gi carbon chik di jar wa imana carbon 2 chi yo chagro tela carbon sum khachi chi yo man mindo tela kharre la wi imana after carboxylation two molecules of 3-phosphoglyceric acid is formed PGA khachu chagre la wi imana PGA molecule ni chagorda and carbon 2 ta cha so yo chung so this is the carboxylation process kyans temba nya okay now next step khare la wi imana reduction reduction ka du kharchi gore there is synthesis of sugar with the use of energy rich molecules like ATP and NADPH which was produced during light reaction rata light reaction ka du page ar swegyo di shine page chi gorda ATP da NADPH di khawala page chi gorla na sugar swela this step khawla yung gore reduction stage ka du chung gor Calvin cycle gi the next khare 3-phosphoglyceric acid chorwa there are two molecules of 3-phosphoglyceric acid these molecules they are reduced with the help of ATP and NADPH ATP the NADPH molecule then so page cheni ATP ki energy tegre NADPH ki energy nyamdola hydrogen de tegre ditteni kharchi gore they produce sugar sucrose or starch soya ko sur thangbo step kharsu do 3-phosphoglyceric acid de machani triose phosphate su do triose phosphate na ne chik kharche chu du re ma starch soya chu du so this is the reduction step and finally third stage dikhare la wi na regeneration the regeneration ka du kharchi gore ribulose bisphosphate la joro carbon dioxide accept chiye molecule cho marwe ribulose bisphosphate la di regenerate chi gore it is regenerated for the cycle to continue kisi ribulose bisphosphate la di tu smart drone glucose soya le pheju chen ribulose bisphosphate de mewa cha wi na plant gi sobu na lo la carbon dioxide accept chingi gi molecule de mewa cha ru goro di mayo ya che du kharchi gore ribulose bisphosphate di regenerate chi gore and during the regeneration process ATP molecule is used the ATP roson rosa chik dela do reduction ka du roson ru do chik wala du gore regeneration ka du roson ru gore regeneration ka du kharchi gore la wi bana from triose phosphate it produces ribulose bisphosphate there is a regeneration of ribulose bisphosphate so that the cycle can continue chilone atmosphere ne carbon dioxide da radigra ine sobu nanglo la plan korangi chloroplast nanglo la ribulose bisphosphate ki supply the yang kare continues to de chedu what it does is there is regeneration of ribulose bisphosphate so these are the three steps very important steps Calvin cycle ki steps sumdi nazo no dia busu nyama do Calvin cycle nanglo la stage sum yore carboxylation reduction and regeneration I hope you are clear regarding this one kiranzo loktim nang remu ditina dir ani Khan Academy la gi website di kala konjo gi na ro stage ribulose bisphosphate kaji molecule di detion da yu gogo den so khari chongore carbon chongor so it is a 5 carbon compound chikni sumshi ngala do so ribulose bisphosphate the phosphate near so the pejeche ani nyamdo la carbon dioxide journey what it produces two molecules of 3-PGA chikni ni do carbon sum che yone molecule ni so so the carbon dioxide da RuBP phasu jare chu marwe carboxylation process chorwa we call it as carboxylation right ngazo dinye den khala me carboxylation line ba carbon dioxide da ribulose bisphosphate jare chu marwe it is a enzyme mediated process tadel enzyme nenga chumbu pejor enzyme nenga chumbu dikhare la wimba na rubisco ribulose bisphosphate carboxylase oxygenase lager the kiranzu full form temba nyayar da you have to remember this right RuBP da carbon dioxide ni di ni carboxylate che chungye de huni ni phazu penju jar chungye de sure la win ba na rubisco lag enzyme jor so this is an enzyme rubisco enzyme ki full form dikha raha hai ribulose bisphosphate carboxylase oxygenase CO la dikha raha hai carboxylase oxygenase so this particular enzyme is very important and especially this enzyme is the most abundant enzyme in the world rubisco la as yema enzyme kab du jayar so 3 carbon compound che jode ni cha so 3-PGA ni ani dende nalo ne chik khwala du do one goes to make glucose swela chik dende carbon sum che jo compound chik ma glucose swela du do ani shimba khawa du gore ngadi archi gore RuBP regenerate chela du so they yago sia din nalo thana glucose chik cha gyur da we have got only half a molecule of glucose over here after fixing 3 carbon dioxide within the Calvin cycle, right? After accepting 3 carbon dioxide within the Calvin cycle, only half molecule of glucose is made. Ta kesi full molecule of glucose so we na thandyo khajo or carbon dioxide sumya thengu waro. Di into zambala, the cycle should run 6 times, right? The Calvin cycle should run 6 times to produce 1 glucose molecule. The Calvin cycle should run 6 times to produce 1 glucose molecule. I hope you remember that. Calvin cycle the reaction should occur 6 times. Carbon dioxide then 2 ya nalo theni ani shine glucose molecule chik swe du. Ajin carbon dioxide sum theni glucose molecule chega swe yo na carbon dioxide 2 theni we mana glucose molecule kangsan chik swe du ro. So D into zambala. How much ATP and NADPH is used for the production of 1 glucose molecule? Glucose molecule chik swewala plant ki ATP da NADPH khazu rosong tango ore lawin by na ata kiranzu calculate chidizama kharche go you have to calculate by using this number over here. Carbon dioxide sum the la khazu do ATP 2 rosong do NADPH 2 rosong do ta del ATP anga dome chigel del sumyor. That means for 3 carbon dioxide, right? For 3 carbon dioxide fixed chidizama la kharche go ore ATP gu thamba rosong roso NADPH 2 thamba rosong roso. Ta kisi carbon dioxide yangya sum the yin by na adi multiply by 2 ta ro ani gu ni gi khazure chopke ATP chopke ta NADPH chungni rosong ro. So I will show you over here. For the synthesis of 1 molecule of glucose, Calvin cycle nanglola glucose molecule chik swela ATP khazur rosong ro re NADPH khazur rosong ro re la yin by na ATP chopke ta NADPH chungni rosong ro. And carbon dioxide khazu ya nanglo thengu gore carbon dioxide 2 thengu gore carbon dioxide ya nanglo 2 thengu me glucose molecule chik swe du re ani ATP chopge da NADPH chopge rosong ro gore. So ATP energy rich molecule di ma cha we men kho kharcha re ADP cha ro NADPH di ma cha we men kharchi ro ko energy rich molecule H di pha ko we men NADP plus cha ro. So this is the overall expenditure of the plant to produce 1 glucose molecule. And Calvin cycle should run 6 times to produce 1 glucose molecule. And with this, I hope you are clear regarding Calvin cycle. Calvin cycle the next C4 pathway C3 pathway clear C3 pathway Calvin cycle when you go to colleges, you will study about the molecular structure of all these intermediate metabolites. RuBP da carbon dioxide carboxylation kabdo ya journey ani khari chagdo 3 phosphoglyceric acid chawara 3 phosphoglyceric acid ki sobda di changor transit to atona and dine ni glyceraldehyde 3 phosphate dela G3P to din sobda ge changor. So all these intermediate metabolites, the structure of which you will be studying about them in your higher classes. Right now it will be sufficient for you to know this particular form of Calvin cycle. Calvin cycle ki simple form di themba tinati wor. Calvin cycle langa zo ming shimba ji kharla gore C3 pathway ke lago. C3 pathway kharchin lago re lago imbana the first compound khar form chi do 3 phosphoglyceric acid, which is a 3 carbon compound, therefore it is also called as C3 pathway. Okay, so before you watch the next video, please read about C4 pathway. C4 pathway kala ya puchine lo ani C3 pathway da C4 pathway kala kewa khardu kuni ni para la khari kewa do rob chi ha kho chi ani shine video de ta ani shine seu cha gre shimba ya dis konyo gu cha durta. Okay, so please read about C4 pathway before you watch the next video. Thank you.